Welcome to Da Vinci Academy's Biochemistry lecture course. I'm Dr. Maxwell Cooper, and I'm excited to be one of the lecturers taking you through this course. We're starting out with Unit 1 here, Molecular Biology. This is the first lecture, and in this lecture we're going to be covering DNA structure and DNA synthesis. Alright, so nucleotides are the building blocks of DNA and RNA, and it starts with the nucleoside. And a nucleoside is a pentose and then a purine or pyrimidine base, and then you form a nucleotide from a nucleoside by phosphorylating that pentose. So then you have a phosphorylated pentose and a purine or pyrimidine base, and that's what gives you a nucleotide. Now the difference between RNA and DNA is that the pentose in RNA is a ribose, so R and R, and then the pentose in DNA is a deoxyribose, so D and D. So the bases, you have adenine, guanine, and cytosine, which are in both DNA and RNA. So these guys right here. Then you have thymine, which is here, which is only in DNA. And then lastly, you have uracil, which is only in RNA. And uracil is actually created through the deamination of cytosine. So you have cytosine, undergoes deamination, gives you uracil. So you have two groups of these nucleotide bases. You have purines and pyrimidines. And they each have a common structure. And you can see that here with the purines first. They each have a fused double ring, which consists of a 5-membered ring and then a 6-membered ring. And the purines consist of adenine and guanine. Then you have the pyrimidines, and they each have a single 6-membered ring, and you can see that here. Thymine has pretty much the same structure as uracil. The major difference is that it has this methyl group here, which uracil doesn't. And remember, thymine is in DNA, uracil is in RNA. So how do you put these nucleotides together to actually form DNA and RNA? Well, that's through a concept called complementary base pairing. And so what that means is you have a purine base pairing with a pyrimidine, and that's actually formed with a hydrogen bond. So these bases will hydrogen bond with each other, and that's what forms double-stranded DNA, and it can form double-stranded RNA as well. And so it has to be between a purine and a pyrimidine. You can't have an adenine base pairing with a guanine or a cytosine with a thymine. It just doesn't happen. So it's always got to be one base from one group, base pairing with another base from the second group. Now, one thing to note, you can see this nice double-stranded DNA structure here. Adenine always base pairs with thymine in DNA, but in RNA, adenine actually binds with uracil. So that's what happens in RNA. Now, guanine always base pairs with cytosine in DNA and RNA. So that's consistent throughout, and you can see that here. Here's guanine base pairing with cytosine. And then when you're forming an RNA transcript, this complementary base pair follows suit as well. And we'll talk much more in detail about this in transcription when you're transcribing DNA into RNA. But you can see that here where it follows the same thing. You have an A for a T, and then you have a U, because it's RNA, for an A here, and then a G for a C, G for C, and then C for G, and then you can see, and so on. One thing to note is that the guanine and cytosine base pair is actually stronger than the adenine and thymine pair. And the reason for that is that the guanine-cytosine pair actually has 3 hydrogen bonds versus adenine and thymine only have 2. So there's more bonds, requires more energy to break it apart. And because of that, let's say you have 2 DNA structures, 2 double strands here. If this has more GCs, DNA strand 1 versus 2, if there's more GC pairs in this one's going to be able to withstand higher temperatures because there's more bonds to break. And then lastly, one thing to note is that DNA is always double-stranded, and that RNA can actually be single or double-stranded. All right, so now that we've talked about the building blocks of DNA, let's talk about how it's actually packaged in the nucleus. So we started with the double helix. You can see that here with the base pairing. And then it's further condensed into a structure called chromatin. And you can see that here in the diagram. And chromatin is actually composed of DNA wrapped around these histone proteins like this, and you can see it wrapping around, and then it gets further condensed into a structure like this, and then chromatin eventually gets further condensed into a chromosome, and that's what you find in the nucleus. Chromatin allows for DNA to fit in the nucleus, and then it also helps with regulating gene expression and DNA replication, because you can unwind chromatin from this densely packed structure like this to a loosely packaged structure like this to allow for these processes to occur. So let's start with the basic structure. First, you have the nucleosome, which is negatively charged DNA. So DNA is negatively charged, and it's wrapped twice around a histone protein, which is positively charged. Remember, opposites attract. And this forms that beads on a string looking appearance. So if you have beads like this on a string, and so on. There's the linker DNA, which connects adjacent nucleosomes, and you can see that here, connecting one nucleosome to the next, and it's associated with the histone H1. So these histone proteins, they have subunits or components, and there's H1, H2, and so on. The big one you want to know is H1 because for this reason, because it's associated with the linker DNA. Then the actual amino acid sequence of histones is rich in lysine and arginine. That's just a good fact to commit to memory. So now euchromatin versus heterochromatin. So heterochromatin is actually best represented by this structure right here. And as you can see, it's condensed chromatin. It's sterically inaccessible to transcription proteins or DNA replication proteins. And as you can see here, it's hard for a protein to get in here. And so you would call that transcriptionally inactive. Now, since it's much denser, it appears darker on electron microscopy, and that's important to know. Euchromatin, on the other hand, is best represented by this structure here. It's much less condensed. It's sterically accessible to both transcription proteins or DNA replication proteins. Hence, you would call it transcriptionally active. And so what happens is that chromatin actually can unwind from heterochromatin to euchromatin like this, and that allows for gene expression. So this is a way of regulating gene expression. That's how you want to think about this. So not only does chromatin help with packing DNA in and so it can fit in the nucleus, it also helps with gene expression and turning genes on and off. And since euchromatin is much less condensed, it appears lighter on electron microscopy. So DNA methylation, this is another process that helps with regulating gene expression. You have cytosine and adenine, which can be methylated. So if we look here on our sample DNA right here, and it typically blocks transcription. And again, it has to do with steric accessibility. So if you have a transcription protein like this, it's going to be harder for it to get in with these bulky methyl groups. However, during DNA replication, same thing, cytosine and adenine can be methylated, but it occurs on the template strand. And what this does is it allows for DNA repair enzymes to distinguish the template strand from the newly synthesized strand. So you have your template, you have your newly synthesized strand. And so what happens is that if there's a mistake in the newly synthesized strand, the repair enzymes want to be able to recognize that. They don't want to act on the template strand. Lastly, there's regions of DNA that contain a cytosine followed by a guanine. And you can see that here in the 5' to 3' direction. And these are called CPG sites. And they're highly concentrated in areas of DNA called CPG islands. So you'll have regions of the long strands of DNA that have a lot of these CG or CPG sites. And again, the cytosines in these CPG islands are methylated to repress transcription. So it's another gene expression mechanism. So histone modifications, this would include histone methylation and histone acetylation. Histone methylation is less straightforward than DNA methylation. It can actually activate or repress DNA transcription. Histone acetylation, now that removes the positive charge from histone. So if you remember, DNA is negatively charged, histone is positively charged. And so by acetylating it, you remove that positive charge. And then that allows the negatively charged DNA to unwind even further, thus allowing for transcription proteins to access the DNA. So once again, just another way of regulating gene expression. All right, so now that we've talked about DNA structure, let's talk about DNA synthesis. And specifically, we're going to talk about the synthesis of purines and pyrimidines. So first with purine synthesis, you have the components that are necessary for synthesis here. So you've got 3 amino acids, glutamine, aspartate, and glycine. And then you also need carbon dioxide and then N10 formyl tetrahydrofolate. And this is a nice diagram here because if you remember, the common structure for purines is a 6-membered ring fused with a 5-membered ring. And then you can see all the starting components here and where their structural contributions are. So this is an important figure to know, and you definitely want to commit this to memory. All right, so this diagram's in your book, and it shows a nice overview of pyrimidine synthesis and purine synthesis, and we'll go through each of these individually. These are good overview diagrams. They don't show you every single step. For both of these, not every step is important. That's not a good use of your time. What you want to focus on is the important steps and the regulation. So the starting products. Now with biochemistry pathways in general, like I was saying, the key things you want to know are starting products, end products, rate limiting steps, committed steps, and then regulation. Those are really the key concepts you want to know. Because if you can understand those, especially the regulation, you want to understand, you don't want to just memorize these pathways. You want to understand where they actually fit in general cellular function and physiology. Now the starting products for purine synthesis are ribose 5-phosphate, which you can see this guy here. and then you have an ATP, which gives you a phosphate group. So you end up forming ADP on your way to synthesizing phosphoribosol diphosphate, or PRPP. And then it's catalyzed by this nice enzyme, PRPP synthetase. And this is the rate limiting step, which is important to know, because what the rate limiting step means is that the entire rate of the pathway depends on this reaction. And this is actually the slowest reaction. So this reaction determines the rate of the overall pathway, hence rate limiting. Then you have the committed step, which is the other important type of step. So on PRP's journey to forming IMP, again, this is an overview diagram, we're not showing you every single step. There's this first step where PRPP is forming 5-phosphorybosylamine, and then it's using glutamine, which is then converted to glutamate, to form that. And it's catalyzed by the enzyme glutamine phosphoribosol pyrophosphate aminotransferase. Now that's a nice mouthful. So for short, we'll call it GPAT. What's more important is the inhibitors and the activators. This is a step on the way to forming IMP, or inosine monophosphate. Now IMP ends up forming adenosine monophosphate, or AMP, and then it can also form guanosine monophosphate, also known as GMP. And we'll go into these two pathways in the subsequent slides. Now all three of these inhibit GPAT. So they inhibit a concept called feedback inhibition, which means if you have a buildup any one of these, it'll come back and inhibit this enzyme. And what that means is essentially just slow the rate of it. On the flip side, PRPP is an activator. So if you have a buildup of PRPP, it's going to activate GPAT and drive this reaction forward. And in fact, if you have high enough concentrations of PRPP, it can overcome feedback inhibition. So if you have a buildup of any one of these three, and then you have a more significant buildup of PRPP is going to win and drive this reaction forward. Now, getting down to the end here, we have IMP, which is converted into AMP or GMP, and there's an exclusive pathway for each of these. Now, you might be wondering, well, which way does it go? It essentially goes both ways, because what you want to end up doing is producing equal amounts of AMP and GMP, because these are going to go on to form adenine and guanine, and you want equal amounts of these. And there's two mechanisms of regulation that control these two pathways that ensures there's an equal number of AMPs and GMPs synthesized. So first you have cross-regulation between the pathways, where essentially one product from one is needed for the other. So first you have the GMP pathway, which actually requires an ATPs from the AMP pathway. So AMP pathway generates this ATP, which is then used by the GMP pathway. Vice versa, the GMP pathway produces a GTP, which is then used by the AMP pathway to produce AMP. So if you shut down synthesis of one, you're also going to decrease this, because decrease AMP, you'd have decreased ATP, and then you're going to lead to this, and then vice versa. And then on top of that, AMP feedback inhibits its own synthesis. Same thing with GMP from IMP. Now you'll notice in your book there on this diagram, we have a box here of drugs that act on these pathways. And so what we're going to talk about is chemotherapy drugs that specifically, in this case, inhibit purine synthesis. Now these are examples of what's called cytotoxic chemotherapy versus the newer drugs in the last 10 or 20 years, which are targeted chemotherapy. So cytotoxic means they just kill, essentially you want to think they kill cells. Usually these are used for cancer. In cancer, you want to prevent significant cell division that's occurring. You want to prevent that, and you want to kill cancer cells. So it's just going to target rapidly dividing cells, because it's going to target cells that are synthesizing large amounts of DNA. And in order to divide, you need to synthesize large amounts of DNA. Versus targeted therapies, these are going to be more so targeted at specific proteins that are molecular drivers for cancer. These are the newer age therapies. Both of them are still commonly used. So in this case, we have these three drugs that we'll talk about that act on the purine synthesis pathway. So first, 6-mercaptopurine, or 6-MP for short. Mechanism, it's a prodrug, meaning the patient takes this what's called azathioprine, which is then converted metabolically in the body to 6-MP. 6-MP inhibits PRP aminotransferase enzyme, which is also known as GPAT, which catalyzes that committed step that forms 5-phosphoribosylamine from PRPP. Remember that we just talked about this a few slides ago. So it's going to act in this part of the pathway here on the way of synthesizing IMP from PRPP. And so this drug is used for acute lymphocytic leukemia, or ALL, and then it's also used for Crohn's disease, and ulcerative colitis, which are autoimmune diseases that cause significant levels of inflammation. So for ALL, you have rapidly dividing lymphocytes in the bone marrow, and so you want to target that. In the Crohn's disease and ulcerative colitis, you also have significant division of immune cells, but for different reasons. These are autoimmune diseases, and their symptoms are caused by significant levels of inflammation that are brought on by significant immune reactions. So you want to taper that immune reaction by decreasing the amount of immune cell division. Mycophenolate and ribavirin, these are two drugs that have the same mechanism but different clinical uses. They both inhibit IMP dehydrogenase, which is a critical enzyme, as you can see in the figure here, from bringing IMP to GMP. Again, the individual steps are not important. It's just important to know that it acts on this enzyme specifically and that it's involved in synthesizing GMP. Their uses: you have mycophenolate, which is an immunosuppressant drug for organ transplant patients. Same concept as autoimmune diseases. For an organ transplant patient, you want to prevent organ rejection by the immune system. So again, you want to prevent rapid division of immune cells. Ribavirin, it's an antiviral drug for RSV infection, hepatitis C, and viral hemorrhagic fever. And then hydroxyurea, so this is a drug that inhibits an enzyme called ribonucleotide reductase. So let's go down here to this. We extended the figure down. So you have AMP and GMP that are then converted into ADP and GDP, respectively. And these are ribonucleotides. And then these are converted by the enzyme ribonucleotide reductase into deoxyribonucleotides, which are DADP and DGDP, respectively. So you inhibit this enzyme with hydroxyurea, which would then prevent the eventual synthesis of adenine and guanine. Uses for this are sickle cell anemia, chronic myelogenous leukemia, or CML, polycythemia vera, and then cervical cancer. All right, so now that we've talked about purine synthesis, let's talk about purine degradation and the purine salvage pathway. And both of these processes or pathways are shown in this diagram in your book. First things first, I want you to correct this. There's adenine and adenine here in your book. This adenine should be PRPP, so make sure you correct that. We apologize about that error. And then the other thing is this part and this part are the purine degradation. It's essentially getting AMP to uric acid and getting GMP to uric acid. That's purine degradation. This part here is the purine salvage pathway. So we're actually going to wipe that part out while we explain purine degradation so that there's no confusion, because otherwise this looks like kind of a convoluted diagram. So that now that we have that whited out, let's talk about purine degradation. So you have excess purines, could be a result of cell lysis. It just could be excess purines within the cell. Cell lysis, the reason why that would be important is, let's say you have a cancer patient with a tumor and you give chemotherapy. It kills a bunch of cells. Then you're having a much more increased level of purines in the blood. Purines get degraded down to uric acid. So what's important to know is they're degraded through a series of steps to uric acid. So this is the main end product here, is uric acid. Someone asks you, what's the end product of purine degradation? Uric acid's the answer. Uric acid then gets secreted in the urine. So if you remember, we'll just highlight this here, even though we're going to focus on GMP and AMP. You have IMP here, which you remember, PRP is converted to 5-phosphoribosylamine, which is then converted to IMP. And so IMP, if you remember what's important about IMP, is IMP can be converted into GMP or AMP. And then this is how IMP gets degraded, and we'll talk about that in a second. All right, so now to focus in on this pathway. So we're going to talk about GMP and AMP mainly here. So AMP, it gets converted into IMP, and then IMP goes down to inosine, then down to hypoxanthine. And we've left out a lot of the enzymes here because they're not particularly high yield. The high yield ones are these ones in blue, and the reason for that is these are enzymes that are involved in a disease or they're acted on by drugs. And so we'll definitely go into that in the next few slides, and especially in the clinical parole section. So then you have hypoxanthine, which is converted into xanthine by xanthine oxidase. And no, this is not a mistake. This enzyme acts on both of these steps. So then it further catalyzes xanthine into uric acid. Then you have GMP, which is converted into guanosine. Guanosine is converted into guanine, and then guanine is converted into xanthine. And then xanthine is converted into uric acid by xanthine oxidase. Again, this is just a slide explaining that xanthine oxidase catalyzes both these reactions involved in uric acid production. And really the important thing with xanthine oxidase is the medications allopurinol and febuxostat, which both inhibit xanthine oxidase, and really their goal is to decrease uric acid production. So why would you want to do that? Well, gout is a disease, as we'll talk about in the clinical pearls section, is a disease that's a result of really increased uric acid levels in the blood. And what ends up happening is uric acid forms crystals in joints and causes pain. And those are what are called acute gout attacks or gout attacks. And so what you want to do is give these drugs to patients who are at risk or have had gout attacks in the past to prevent future attacks. The other thing you want to do is prevent tumor lysis syndrome, for example, in like a cancer patient that's receiving chemotherapy. You kill a lot of cancer cells at once with chemotherapy. You could have a dramatic spike in uric acid, which could then lead to tumor lysis syndrome. So it's used for that. And then it can also be used for add-on therapy for patients with inflammatory bowel disease that have not responded to monotherapy for that disease. Probenecid, this is just for completeness sake. It's not involved in any of these processes at all. It's just a gout medication that works by blocking reabsorption in the kidney. So essentially, it increases uric acid secretion in the kidney or in the urine. All right, so now that we've added this part back in, this is the purine salvage pathway. The key enzyme here is hypoxanthine guanine phosphoribosol transferase, or HGPRT, and you can see that here. This is an enzyme. It's in blue like the other enzymes. Don't get confused. It's not an intermediate. And it's involved in two different salvage reactions, or de novo synthesis of purines. So you have guanine and hypoxanthine. Converts hypoxanthine into IMP. That's important because IMP can go to GMP or IMP. And then it converts guanine into GMP. And what's important to note is that PRPP is the source of ribose 5-phosphate for both of these salvage reactions. And remember, ribose 5-phosphate is actually one of the required starting products for purine synthesis. Now, you may be wondering, what's the purpose of this? Why even do these salvage pathways? Really, the main thing is you want to decrease uric acid production, as you saw on the previous couple slides, why that's a problem. And so this is kind of the body's own way of essentially recycling excess purines. So you could think of it that way. So there's definitely purines that are going to get converted into uric acid, but you're not going to convert all of them. You want to recycle them back to GMP and IMP so that then you can eventually create more purines. Adenine is salvaged through a separate pathway that also uses PRPP as the source of ribose 5-phosphate and eventually yields AMP. So you have PRPP combining with adenine to form AMP, and this is catalyzed by the enzyme adenine phosphoribosol transferase, or APRT. That's not typically high yield enzyme to know about. It's just that you don't want to get confused and think that HGPRT is also responsible for salvaging adenine. Adenine has its own enzyme. So with pyrimidines, the big thing you want to know is it's not degraded to uric acid. It has its own 3 end products, which are ammonium, beta amino acids, and then carbon dioxide. And the big thing to take away here is the ammonium is then used by the urea cycle, which we'll talk much more in detail about in the metabolism unit, to form urea, which is then excreted in the urine. All right, so now we'll talk about pyrimidine synthesis. Now first, the components that are necessary for synthesis are aspartate and carbonyl phosphate. And you see we have this nice diagram here showing you the common structure again, 6-membered ring, and you have aspartate and carbonyl phosphate and what their structural contributions are to that 6-member ring. Again, just like for purine synthesis, this is an important diagram to commit to memory. So the synthesis pathway, again, this is an overview pathway. It does not show every single step. Again, knowing every step is not important. You want to know the key steps and then the key regulation. So the starting products, this is a good fact to know: glutamine, CO2, and ATP. You can see these guys here, glutamine, CO2, and then ATP will come in here and produces an ADP. And then what this reaction does is it forms carbonyl phosphate, which is catalyzed by the enzyme carbonyl phosphate synthetase 2, or CPS2. And this is in the cytosol, which is important to know. There's CPS1, which is in the mitochondria. It's a different enzyme, different function. And the regulation here is you have uridine triphosphate, which is an example, again, of feedback inhibition. To the downstream product comes down and inhibits this main enzyme, which is involved in the committed step. Again, this is important. So once you form this step, you're committed to forming pyrimidines. It's activated by ATP and PRPP. Now, PRPP, you remember us talking about it in purine synthesis. It's a molecule that's involved in both pyrimidine and purine synthesis. and its function is to transfer phospholipase groups for the synthesis of several nucleotide monophosphates, such as GMP, like we talked about, AMP, and then in this case UMP, and so on. So this is involved in both. So if you have a high level of this, it's going to drive, basically you want to think of high level PRP is going to drive both purine synthesis and pyrimidine synthesis. So we go down the pathway here. You have our aspartate. Remember that starting product comes in, helps form orotic acid. Then you have UMP, UDP. Then you have this ribonucleotide reductase coming into play again. Remember we talked about this in purine synthesis, so specifically hydroxyurea inhibits this. So this catalyzes the conversion of these ribonucleotides to deoxyribonucleotides. And then you come down here to this portion of the pathway, which is really high yield because it has a couple enzymes that are targets of high yield drugs. So you have thymidylate synthase, which actually catalyzes a methylation reaction. And so it catalyzes the addition of a methyl group from N5, N10 methylene tetrahydrofolate, or THF. So it catalyzes an addition of CH3 to here to then form DTMP, deoxyuridine monophosphate, DMP. Now, N5, N10 methylene tetrahydrofolate, this is formed from THF, or tetrahydrofolate, which is then formed from dihydrofolic acid, or DHF, by dihydrofolate reductase. And so folate metabolism is intricately connected to pyrimidine synthesis and then hence DNA synthesis. And we'll talk much more in detail about folate and its function and metabolism in the nutrition lectures. So now a couple chemotherapy drugs that inhibit pyrimidine synthesis. So first, 5-fluorouracil, or 5-FU. This is actually a pyrimidine analog, which means it's structurally similar to pyrimidine bases. And it's actually a prodrug that's enzymatically converted to 5-fluorodeoxyuridine monophosphate. So this patient is given this, and then it's converted in the body to FD-UMP. FD-UMP actually forms a covalent bond with folic acid, which then irreversibly inhibits thymidylate synthase. So you inhibit this enzyme, you decrease formation of DTMP from DUMP, and then importantly, the end result, you decrease DNA synthesis. This is going to target cells that are rapidly dividing, and if cells are rapidly dividing, they need a high level of DNA synthesis. So these are the cells that are going to be most affected by 5-FU. And as a result of that, 5-FU is a chemotherapy used in a wide array of cancers. Methotrexate is a folic acid analog that competitively inhibits dihydrofolate reductase. So it is structurally similar to folic acid, and then it inhibits dihydrofolate reductase at the active site. So then you have decreased formation of tetrahydrofolate, decreased formation of N5, N10 methylene tetrahydrofolate, which then decreases that methylation. So you're going to have decreased formation of DTMP, which then as a result decreases DNA synthesis. So if you can see, it actually has kind of the same end result as 5-FU, it just acts on a different enzyme. So acting on folate metabolism indirectly inhibits pyrimidine synthesis. And methotrexate is also used for a lot of cancers. It's also used for inflammatory disorders such as rheumatoid arthritis, and it's used again kind of like what we talked about with other drugs that act in inflammatory disorders for purine synthesis. Same thing, you want to decrease inflammation, so you want to decrease immune cell division. So it's the same kind of concept coming into play. All right, so now we're going to talk about some clinical pearls relevant to DNA synthesis. So first, gout. You've heard us talk about this a lot. Essentially it's a form of inflammatory arthritis. You got to keep that in mind. Even though it has a biochemistry base to it, it's just like osteoarthritis or rheumatoid arthritis in the sense that it eventually causes joint destruction and joint pain. But it's characterized by high levels of serum uric acid, as we've talked about. And this is caused by either on the front end overproduction of uric acid or under excretion. So it's either a problem at the biochemistry level where you're overproducing it, or on the renal side in the kidney where you're not excreting enough uric acid. Now, what characterizes this is a disease that occurs over time. These patients, they have chronic elevated levels of uric acid. And what this results in is monosodium urate crystal formation in the joints. And so these can affect really any joint in the body. So what happens is these crystals form in the joint, they harden, and then they bang up against other structures, specifically the cartilage, and then they trigger a lot of inflammation. And what that does is ends up causing the symptoms, which are significant pain in these patients. A key fact to know is the metatarsophalangeal joint of the big toe is the most commonly affected joint. And so you can see that in the x-ray here. There, you can see these crystals, significant amount of inflammation. It's affecting both of these joints here. So really the mechanism here, you can have either overproduction or underexcretion like we talked about. We'll focus mainly on overproduction since underexcretion is more at the kidney level. So you have overproduction of uric acid. This can be a result of really one of two things. So you either have overactivity or PRPP synthase. Now if you remember, AMP, IMP, GMP, PRPP plays a key role in production of all of these. So if you have an elevated level of PRPP, you're going to have elevated levels of these, and then as you can see, these all eventually funnel to uric acid. The other example is you could have deficiency of HGPRT or absent purine salvage. So if you knock this out, remember we said the purpose of purine salvage is to keep the levels of uric acid normal and keep them from getting elevated. Versus if you don't have this or it's slower or deficient, you're not saving enough of these to then be recycled and produced into purines again. And so you're going to have, again, increased levels of uric acid. The other thing is cancer patients that are receiving cytotoxic chemotherapy, those drugs that really focus on rapidly dividing cells, they result in killing a lot of cells. And so what happens is you have high levels of uric acid in the blood, and this results from high amounts of cell lysis that are essentially dumping these purines into the blood. This is what's called tumor lysis syndrome. You can see this in a variety of different cancers, but commonly you'll see it in cancers such as leukemia. And this is a very important concept to understand. This is a very high yield topic for board exam questions. And then on the back end, just for completeness sake, you have underexcretion, which is really caused by defects in renal excretion of uric acid, and that's more of a physiological concept. Some risk factors, these are just key to know. These could come up on a test question for sure. Patients that consume a lot of alcohol, sugar, and or meat, especially red meat. Obesity, which is not surprising. If you consume a lot of these things, you're definitely at risk for becoming obese. Goes hand in hand. And then also medications that patients take, as we've talked about a lot, chemotherapy, and then diuretics, which are more on the renal side, because diuretics act in the nephron of the kidney, and so they're more so on the result in underexcretion of uric acid. The presentation, like we talked about, is really significant pain. These patients are really uncomfortable, really in a lot of pain. And remember, the sole cause is inflammation. So swollen, red, warm skin around the joint, these are all signs of inflammation. You've probably seen this before as a classic painting of a gout attack. And then here's a nice picture of it showing these, that red skin, warm, very swollen joint. So how do you diagnose it? Obviously, they're going to have elevated levels of uric acid. But more importantly, you're going to get an x-ray and you're going to see something like this where there's crystal formation, a lot of inflammation. And then really the way you confirm the diagnosis is you're going to tap the joint, get synovial fluid, and analyze it. And what's going to show is monosodium urate crystals, those crystals that form as a result of high levels of uric acid. And they're going to be negatively bipheringent under polarized light. That's how you confirm that these are, in fact, these crystals, and that's how you confirm gout. Here's an image of this. This is a good image to commit to memory. You're going to see this a lot in Q-bank questions, and you could definitely see it on a board exam, and so you want to definitely associate this with gout. So treatment, acute therapy, these are what's called the gout attacks. So how you treat those when a patient really comes into the hospital or the doctor's office with these acute attacks. You're going to give them NSAIDs or colchicine. This is a common question when they try to trip you up. You're not going to give them something like allopurinol. That's for long-term therapy. So you're going to give them NSAIDs or colchicine. You would give someone colchicine if they have a history of peptic ulcer disease or if they have a history of renal disease, because NSAIDs really affect renal function, and then they can also exacerbate peptic ulcers. So if someone has a history of ulcers, they're already in a lot of pain. You don't want to flare up their ulcers. And then if they're someone with kidney problems, you don't want to further complicate those. So you'd give them colchicine, both effective drugs. Now, once you have the gout attack controlled, you want to give them therapy to help prevent these from happening again. So prophylaxis involves medications to decrease uric acid levels, which makes sense, mainly allopurinol and febuxostat, and then you can give probenecid, which acts on the back end and the kidney. And remember, these two drugs, they inhibit xanthine oxidase, which is then going to decrease levels of uric acid production. All right, so adenosine deaminase deficiency. This is an interesting disease because it involves both biochemistry and immunology. We'll focus first on the biochemistry aspect. So adenosine deaminase, just to remind you, it helps degrade adenosine to inosine, which then goes on to hypoxanthine and then xanthine and then uric acid. Big thing here is buildup of DATP. So what you really, for the purpose of this, so you have DATP here. It gets degraded into DADP, which then gets degraded into DAMP, which then gets degraded into, really you want to think of it as deoxyadenosine. So it can, the adenosine deaminase can degrade adenosine or deoxyadenosine. If it's deoxyadenosine, it's going to be deoxyinosine, and then hypoxanthine, xanthine, and uric acid. So what happens is if you knock this enzyme out, you're going to have significantly high levels of deoxy-ATP. This builds up. This negative, since this is an end product of DNA synthesis, it's going to lead to decreased synthesis of nucleotides, which would be negative feedback. It makes sense. If there's enough of the end product, you're not going to produce any more of it. So then the other problem is you have an increase of DATP. This buildup, in addition to decreasing synthesis of nucleotides, it's also toxic to lymphocytes. And so it results in significant cell death in lymphocytes. And that's what ends up resulting in these patients becoming immunocompromised. So the inheritance, for all these genetic diseases, we're going to be sure to point out the inheritance pattern, because this may not seem important, but at least in the short term, for your purposes, these are frequently asked on board exams. They could even be asked on your class exams as well. And a perfect example is that you could have a classic presentation of someone with adenosine deaminase deficiency, and you could certainly know that, and you could know the immunology and the biochemistry inside and out. And the problem is the question could ask simply, what's the inheritance at the end of a long stem? It could just ask that. And if you don't know, it doesn't matter that you know that biochemistry and immunology so well, you're going to get the question wrong. And unfortunately, that's just kind of the way the game's played, whether that's right or wrong. And so unfortunately, you just got to play the game. So you definitely want to be paying attention to these and committing these to memory for these diseases. I know it's not fun, but it's just one way to pick up some more points on the test. Anyways, I digress. What this causes is autosomal recessive severe combined immunodeficiency syndrome, also known as SCID. And these patients, they'll have decreased B cells and T cells. That's important to know. So when you're comparing to other immunodeficiencies, some could have just decreased B cells or just decreased T cells. So that's important to know. We won't get into that because that's more immunology. Clinical features. Now, B cells and T cells, as you'll learn in immunology, these are major league components of the immune system. You don't have these and you're going to be severely immunocompromised. And so what happens is these patients are going to be subject to recurrent infections, very severe infections, especially pneumonia, GI infections. The reason for that is the respiratory tract, you're constantly breathing in air, constantly exposed to pathogens. GI infection, you're eating and drinking, because it's part of basic survival. Again, you're exposed to outside pathogens. And then viral and fungal, they're just not as well equipped to fight off these infections. So how do you treat it? You can give a bone marrow transplant, essentially give them new lymphocytes that don't have this mutation, and then that's one strategy. Another strategy is enzyme replacement therapy, so you give them adenosine deaminase, and then you can give them immunoglobulin therapy. The thing to keep in mind is since this is a inherited disease that starts at birth, these are going to be typically very young children, unfortunately, that will be presenting with this disease. All right, lastly here, Lesch-9 syndrome. So this is another very unfortunate disease. These patients are deficient in HGPRT. If you remember, this is the purine salvage pathway enzyme. Takes guanine, converts it into GMP, takes hypoxanthine, converts it into IMP. So what happens is, remember, the purpose of purine salvage is to prevent increased levels of uric acid and then also provide new substrates for purine synthesis. And so what happens is if you have a deficiency in this enzyme, you're going to have very significant levels of uric acid all the time, much higher levels than the typical gout patient. So the inheritance, X-linked recessive, so typically young boys. Clinical features, obviously they're going to have gout, but then they're going to have, like I said, chronically much higher levels of uric acid, and that results in other symptoms such as orange diapers with sodium urate crystals. They're going to have central nervous system deterioration, retardation, cerebral palsy, dystonia. As you're noticing here, elevated levels of uric acid has a significant effect on the nervous system. And then unfortunately, the hallmark symptom is self-mutilation, which is also a result of damage to the nervous system. So just characterized by nail biting, hitting themselves, aggression. A lot of times these patients even have to be restrained, unfortunately, to keep themselves from hurting themselves. And then unfortunately, these patients, there's no definitive cure, so often early death is the outcome, unfortunately. What you treat is you want to try to control these uric acid levels, so you give allopurinol and febuxostat. All right, and that closes out our discussion of DNA structure and DNA synthesis. Next, we'll talk about DNA replication.